Today we're going to be talking about incomplete dominance. It's another type of genetics problem. There's not a ton you need to write in your notebook. If you want a sample problem, you're more than welcome to write down the sample problem. We're going to talk a little bit first, though, about what incomplete dominance is. So in our normal dominant and recessive problems, like we did previously, we have a dominant gene and a recessive gene. Dominant, we use a capital R, recessive is lowercase. And as long as they have one dominant gene, they're going to end up being the dominant phenotype, red. If they have two recessive, then they're going to end up being the recessive gene color, which is white. In incomplete dominance, we don't have a dominant and recessive gene. You'll notice we usually just symbolize them with two different letters. We don't want to use uppercase and lowercase because one of them is not really dominant over the other. So in this case, if we have two R's, we have red. Two W's, we have white. And if we have one of each, they blend to create pink. So the big word you want to think of when you think of incomplete dominance is blend. Okay, the heterozygous phenotype here, the one that has one of each letters, is always going to be a blend of the other two. So let's do a problem. So it says flower color is incompletely dominant, so it'll always tell you that. When a red and white cross, pink flower results. Cross a pink flower with a white flower. So the first step we always do is determine which trait is dominant and recessive. In incomplete dominance, none of them are dominant and recessive, okay? They're not dominant over one another. So we're just going to skip this. So always on step one, you're going to skip it in incomplete dominance. Step two, determine what letters will represent the genes. So just like in our example, remember, we're going to use two different letters. So it says for red, so we'll use a letter for red, and then white. And then the blend is going to be a combination of those two. So in this example, red, we would have big R, big R, white, big W, and then pink is a blend, R and W. So the next thing we have to do is determine the parent gene. So go ahead, pink flower, white flower. I don't care which is male, which is female, parent one, parent two. One of them is going to be pink, one of them is going to be white. So the pink one's going to be RW because that's the blend, and then the white one's going to be WW. Then we determine the genes going into the sperm and egg, so we just divide them up. Remember, each sperm or each egg only gets one letter. Okay, meiosis creates cells with half of the genes. Then we put them in our Punnett square. Reminder, it does not matter which side they go on. So if you want to put RW up here, great. If you want to write it WR instead of RW, totally fine. The only rule when it comes to putting these letters around the Punnett square is that parent one's letters have to be on the same side, parent two letters have to be on the same side. So that means this W and this W have to be on a side together. Doesn't matter if it's on this side or the top, they have to be together. And then we fill them in, and we determine what our results are going to be. So red, remember, is RR. We don't have any RR. Pink is RW. Okay, you'll see we have two there. And then white is WW, and we have two there. So we determine the offspring, and that's it.